satisfied, you get a quiz drop, and even if you're satisfied with your quiz grades, I would still take it, just because it'll help you know if you're understanding kind of what we're going through. And so, just so everyone kind of has an even footing going into the quiz, it's going to be PCR, right? So that's what you should be expecting, something about PCR. Probably not drawing PCR, but probably about primers and being able to think about primers and polarity of DNA molecules, which will then help prepare you for exam 4. Other announcements, peer review is due next Monday. I believe if you're struggling finding them, I sort of know where they are, but the best resource would be other students. So if you can't figure it out today, come to discussion, ask someone to help you, or we can try to do it together. The reason I can't see it very well is because I didn't assign myself peer reviews, so I can't find exactly where they are for everyone. Any questions on any of that? So today we're going to go into epigenetics and then developmental genetics. So again, this section of the course is supposed to be synthesizing. We've done all of these things with probabilities, with kind of different mechanisms of gene regulation, different types of mutations, and now we're synthesizing them. So we won't get to your discussion questions tomorrow. Two are about development, I guess. One is about gene regulation. The first one with the cavefish, the second one with Arabidopsis flower model. That one, the first two you can kind of logic out just based on the model I gave you. The second two you have to look at the book. There's no really way to memorize the specific cases. So put the answer down and then we can talk about it. I forget what the other two are. But anyways, you won't see the Arabidopsis model today, so you have to look at the book. You'll see it Thursday in developmental genetics. Okay, so epigenetics. So my goal here about epigenetics is to try to describe mechanisms and rules. When epigenetics were first being described, a lot of people, not scientists, but a lot of non-scientists were like, oh, it's this magical thing, right? Epi means on top of genetics. It's all of these principles that don't follow genetic rules that we have established. And that's not true. So that's the first kind of thing I would just want to clear there with. It's a misconception. They have rules, they have mechanisms. We might not understand all the mechanisms, but we can make logical experiments and design experiments to test mechanisms. So this is a really famous example. It's about the agouti gene in mice. So the mouse on the right-hand side is a wild-type mouse. The mouse on the left-hand side is an agouti mouse. Agouti controls coat color, but also has this really interesting effect on methylation patterns and diet. So you get all these interesting obese phenotypes iotropically linked to agouti, right? Because agouti is not involved in metabolism directly, but how it's involved in methylation and therefore kind of have downstream effects on a whole organism phenotypes. Agouti was really a popular case study because we know that there's epigenetic effects in humans. So I don't know if any of you, this is the example from your book. I don't know if any of you have seen it previously, but it's this really famous example about kind of if you stress humans through starvation, what happens to metabolism in subsequent generations. So this was called the Dutch hunger winter. So after World War II, severe famine in the Netherlands. It wasn't just children who were conceived during this time that had effects, but also people have done further studies with actually grandchildren too. So how is that happening? How is a signal from the environment, famine, being stably transmitted through generations? Methylation, right? Epigenetics. They follow rules of inheritance that we've been talking about, but the probability is different because the way that we inherit methylation marks are different. So what is epigenetics? So the most general definition would be how a genotype produces a phenotype, right? That's what we've been focused on this whole class. And then we would just add on through development. Epigenetics and developmental genetics can be partitioned a little bit because there are plenty of developmental traits that are not epigenetic. They're just straight Mendelian mutations. But I think most people, epigenetics as a field, would define it this way. And it really is kind of a combination of the two words. So epigenesis is how an embryo develops. And then we know genetics, right? Study of genes and heritability. So when you combine these terms into epigenetics, that's what I want you to focus on, that these changes are heritable. You don't have to memorize these definitions, but hopefully if you see where they come from, it's not a mystery. Genetics is heredity, so epigenetics is still going to be a heritable change. So you can think about patterns and consequences for phenotypes. So there are many different types of changes that kind of are classified in this larger umbrella of epigenetics, and some that are not. So here's our first top hat question. Which of the following is not a mechanism of epigenetic change? So most of you have answered. One answer you guys all ruled out, DNA methylation. So that's great. That's what we've been kind of talking about so frequently, and I used in the example kind of starting class. So DNA methylation is an epigenetic process, or is a mechanism that can lead to epigenetic change. So we'll rule that one out. What about the others? What do you guys want to rule out? Someone over here, louder. Yeah, we can rule out histones. Histones are really similar to DNA methylation, right? It's a change. It's not happening to the nucleotide sequence. It's happening to kind of histones. But remember, histones are forming complexes with DNA and chromatin. So it's the same idea. It's about accessibility, right? Or adding a functional group that's going to change how proteins interact with the chromosome. So I think the logic is the same way that DNA methylation is going to be an epigenetic process, so is histone acetylation. It's a mark, something in the genome distinguishing that region as a mark, but that's all it is, it's a mark. Okay, so we rule out those two. What about B and D? These are a little more tricky because they are actual kind of molecules that we talk about a lot in normal genetics, right? DNA and RNA.
Rule out D.
And why?
I just remember the list before.
Yeah, so we can rule this out two ways. We can think of, a change of the DNA sequence is not going to be epigenetic, so that gives us the best answer B. But we can also think about D. Well, why not D, right? We have DNA changes, they're transcribed, they're translated, so RNA molecules are doing something. We'll see an example in activation. What else can RNA do, right? They can actually interact with DNA through complementary base pair interactions. They can inhibit, they can get in the way of accessing DNA molecules through binding to them. So that's one reason you can memorize what the book says. You can just think about it. The RNA that we see in genetics is a template. This is describing an active role of RNA. So it would not fall under genetics, so it would fall under epigenetics. Any questions on that? We'll see an example of that with X and activation. Transcript is transcribed, it kind of coats the X chromosome, and it makes that transcript, or that chromosome no longer transcriptionally active. So epigenetics can have kind of really fundamental changes, or they can fundamentally change the nature of what we might see as a phenotype. So this is another example from your book, Linearia. Same genotype, just different epigenetics. Differences in DNA methylation or histone acetylation gives these really variable phenotypes here. So this would be the wild type flower, and this is called a caloric mutant. And they only differ in DNA methylation. You know they only differ in DNA methylation. Sequence the genomes, they're identical. Or start with stocks, right? True breeding stocks. We've seen that from a really, really long time ago. And do something. Change methylation. right? Use all those genetic techniques we were talking about to think about how you can change DNA methylation. You can do an experiment and demonstrate that this is just due to methylation genes. So we've seen this previously, this idea here as well, when we were talking about gene regulation. So at that time, we weren't talking about it in terms of epigenetics, but they're synonymous. When we were saying, here's a bunch of mechanisms that lead to gene regulation changes that are not changes in kind of the regulatory regions, we were talking about epigenetics. So now we can just hash them kind of formally like that. So what do we have? DNA methylation. Chromatin structure. And then part of this could be chemical modification of histones. So this is where we saw acetylation. And then RNA molecules, but specifically ones that are going to kind of affect chromatin. Not templates. Not templates for transcription, something else. And again, what are these all doing? They're influencing gene expression. They have to, by definition. How we define it is connecting genotype and phenotype through development. What's happening through development? All cells have the same genome, they're just expressing genes differentially. So that's the logical connection you need to make. Each of these affects gene expression. Gene expression will affect a phenotype. So that's what we saw last week when we were thinking about the polar bears. That's what you'll see this week in discussion. So the very first one is asking about kind of those blind cavefish that relate to the article you read. That's what you're going to see on the exam. You're going to see data about methylation. You have to tell me about phenotype. So what does DNA methylation look like? Right, so we have kind of our normal bases here, cytosine, you add a methyl group. So if you're just kind of, when you're saying methylation, it really means a physical change in the DNA, but not the base pair change. Normally, this base kind of has what? Saturated carbons. They have hydrogens on them, and now we have this new methyl group. They're accessible, so the part of this picture is like they're accessible. What I really mean is they're on the outside of the DNA strand. Right? If there's the hydrogen bonds on the inside, the phosphate backbone on the outside, these are on, attached to the nucleotides, the bases that would normally make hydrogen bonds, but the way that the changes occur, they kind of are accessible, or they're kind of, the three-dimensional shape puts them on the outside of the molecule. So other proteins recognize that these sites are methyl. Any questions on that? What sites are often going to be methylated? We saw this a while ago too. So we said CPG sites, so C's that are a certain distance away from G's. These dinucleotides are often methylated, preferentially methylated. If you have large stretches of these CPG methylated, they're called CPG islets. So we've seen that term before. So how are these transmitted? How is DNA replicated in a semi-conservative model, right? We saw this in the very first or second lecture in class. We also saw it in PCR. You have an old template strand, you replicate a new strand. That's a semi-conservative model. Each round of DNA replication of PCR isn't just making new strand on new strand. You need a template. You need to build 5 to 3 prime. That's the exact same thing that's going to dictate how methylation marks are passed through generations. So this is the example we've seen before for DNA replication. We have our replication fork. We've only seen part of it. The key thing here is that this is where the semi-conservative model comes in, right? So the old strands have the methyl marks. And then that methyl transferase enzyme is recognizing that and adding methyl groups to the new strands, right? So down here, now both strands have the methyl mark. Or you can say R-methyl. Does that make sense? That's the super important key take-home point of this slide. Semi-conservative replication leaves kind of two new strands. One copy, the gray copy, has methyl marks. This can be used to mark the new strand. So this is in DNA replication, but similar things can happen in when a zygote is formed, right? Instead of thinking about new strands being synthesized, think about haploid genomes coming together during fertilization. Maybe the gray strand is from the paternal, the sperm parent, XY parent, and the red strand from the XX parent, egg parent. If you bring those together, how are the methyl marks gonna be transmitted then? That's what we'll see in a couple slides called imprinting. The methyl marks are not just always added on to every DNA strand. It's true in DNA replication, but in embryogenesis, during the zygote formation, it depends. We have different imprinting. It might depend on whether that allele came from the X parent, the egg parent, or the XY sperm parent. This methyltransferase enzyme is really important. If it's dictating where methyl marks are occurring throughout development, if you have mutations in that enzyme, you can see, or just variation in that enzyme, you can see really different phenotypic results. So the example that your book was using was honeybee development. So, right, honeybees have a social system where they rely on differentiation of workers, and that's due to an environmental cue. These workers and the queen are often, well, the workers are all not identical as we thought. Some queens actually are multiply inseminated, so they're only like half siblings, but the dogma used to be that all workers are genetically identical. They're kind of, some of them are diploid, but a lot of them are these haploid products that the queen makes. So DNA is the same, we'll just assume for simplicity. So how do you get these drastically different phenotypes? This has to do with an environmental cue.
Royal jelly.
That's kind of a recurring theme that we see in epigenetics. Right, gene expression can be in response to the environment. How are you going to change gene expression? You can do it through epigenetic processes. So this is the example from your book. So bee larvae are developing in the hive, in the honey hive, in the honeybee hive, and then there's a signal. So here the signal is the production of this royal jelly. So there's two paths. It can be a worker or a queen. So if there's no royal jelly being provided, there's no activation of the methyltransferase. So that's what this thing here is. That's the DNA methyltransferase. And it's an enzyme that has that ACE. What is its job? To transfer methyl groups onto DNA sequences. So royal jelly, I don't know if we know how this happens yet, but people have done experiments, like feeding experiments showing this. So I think the link between DNMT3 activity and royal honey is not known, but kind of downstream consequences of that. So if DNMT3 is active, producing methyltransferase, the locus is methylated. So it says gene for queen characteristics. It doesn't have to be a single gene, it can be any locus. Basically, a methylation, remember, inhibits transcription. So that's the left-hand side. Royal jelly activates this locus. It methylates DNA, no transcription. Any of the genes that are required to develop into a queen are inhibited indirectly through methyl jelly, directly through DNA methylation. On the other hand, if royal jelly, and so they say or siRNAs, so that's the experimental part. They were showing that you can inhibit the transcription of DNMT3 exogenously, right? This is an experiment. They made a plasmid. We talked about this last class with those monkeys and the cholesterol. They made a plasmid that expresses small RNAs. They injected small interfering RNAs into the gene targeting DNMT3. They see the same result. So DNMT3 is inhibited, less methylation, you get expression of these genes, right? So if there's no methylation, that leads to expression. This sounds really similar to me to like a lactoferron process, right? There's an environmental signal, the presence of lactose, you get different transcription. There's the presence of royal jelly, you get different transcriptional effects. Let's say we're in a hive that has a mutation at DNMT3 that makes it a null mutation, non-functional mutation. What's the phenotype?
It'd be a queen bee.
What's the phenotype if we have a DNMT3 null mutation?
Queen bee?
Always queen. In the presence or absence of royal jelly, you always get queen bees. Does that make sense? You guys see where that's coming from? You can look at the pathway, exactly like we looked at lactose metabolism, right? It's kind of like the super suppressor or like a mutation in the promoter. On the right-hand side, DNMT3 is already not working. It's not being transcribed. So if it is being transcribed, but it's a null mutant, you get both queens. Okay. Those are the kind of logical steps that we can take now, that we have all these tools. That's what I'm expecting of you guys to do. Here's a methylation pattern, here's a genetics problem, predict the phenotype. Any questions on that? Okay, so there's a couple more slides on epigenetics, and then we're going to switch to development. So I already sort of answered this, but this is to try to tie this together to get our brains thinking about these CPG sites. So which of the following is true for the CPG island? So you can rule some of them out based on definition, but then it leaves you with a couple that are just different in direction. So then you have to think about direction. Okay, so answers are coming in. Let's rule out C. Why can we rule out C?
Acetylation.
The definition of acetylation.
So what, or another way to say it, what's being acetylated? Histone. DNA is not being acetylated. DNA is being methylated. Histones are being acetylated. This is describing, a CPG island is describing changes in two nucleotides, right? Cytosine, guanine, dinucleotide pairs. So we can rule out C. Acetylation cannot happen on these types of islands because that's happening on histones. Okay, what else do you guys want to rule out? Kind of rules out D too, right? So what about A and B? They're kind of opposites, right? They're either methylated, near promoters that are active, or they're unmethylated, near promoters that are active. So what does methylation do?
Inhibits
It inhibits.
transcription.
So that rules out A, right? If you have a bunch of methylated DNA, you're not going to be an actively transcribed gene. That leaves you with B. Unmethylated, the absence of methylation groups transcribes genes, right? We saw that in the honeybee example. The absence of methyl on the right-hand side, the absence of methylation groups transcribe genes for queen. That comes from the logic that methylation inhibits transcription. Those are really, really key points. So if you have to start somewhere in any problem with methylation, start there. Start with like your fact. Methylation inhibits transcription, then go somewhere else. Use the rest of the information provided in the question. Questions on methylation phenotype? Okay. How do we detect this? So we're going to try to connect back to sequencing. Because that's really important, right? We need to understand, if we're saying there's only changes in methylation groups, we need to be able to detect them, make hypotheses. So what are some ways we can detect methylation? You might not know, and that's okay, but just like, what are the ways that we detected normal DNA sequence changes? Did any of them possibly apply to these methylation changes? So what did we do to DNA sequences to identify SNPs or identify changes? We did two things. We talked about two things. Yeah, we did gel electrophoresis, great. I like that idea. What are we seeing on the gel though? We can do two things with this gel or something else, yeah. That's a good starting point. Cutting, yes, we can cut things. Okay, what's the second thing we did with DNA? We run it on a gel, we do PCR, but what else can a gel tell us? It can tell us length, but what else? So think about the dideoxy reaction. What were we doing there?
Determine the sequence of genes.
Yeah, we were sequencing things. We talked about sequencing kind of on a single locus or small DNA basis, and then we scaled it up to kind of next generation Illumina type sequencing where all that's happening in parallel. So it's incorporating kind of labeled nucleotides. So we can cut DNA. How would that work here in this case? Most of the restriction enzymes we cared about were recognizing specific DNA sequences, right? Like 4 bases, 6 bases. What needs to happen for restriction enzymes to detect methylation? I know it's early, and I know you only have 2 weeks left of class, but you have all the tools, right? You can do this logically. DNA is going to be identical, so DNA sequence is not going to help us. These methyl groups are on the outside of DNA. We're going to cut something. So what must be true about these enzymes? Okay, we'll get back to it. What about sequencing? What are we going to do with sequencing? The DNA sequence that these two genes should be identical. How are we going to sequence them? Say that again?
The cytosine can be turned into, if it's methylated or unmethylated, it can be turned into or shows up as a reversal.
Yes, exactly. So we'll see a slide on that, but that's what we're going for. The DNA is the same, it differs in methylation, we can leverage that. We can do a chemical reaction. So this is called bisulfate sequencing. So it's a chemical reaction that's going to convert methylated cytosines. If we think CPG islands are methylating these cytosines, we can do chemistry. We can find a chemical or a set of enzymes that's going to see these, change them. Then we sequence the DNA without this treatment. We sequence the DNA with these treatments. Any changes are the cytosines that were methylated. We'll see a slide on that. Let's go back up to these cutting things. So in Papa chats, someone put endonuclease. So endonuclease is removing. I like the idea. It's thinking something has to be happening. The definition of an endonuclease is you're removing nucleotides, or I guess that's exonuclease. Any other ideas on this restriction enzyme? It has to be methylation sensitive. Right, these are kind of linked. We're taking advantage of the fact that some of these nucleotides are methylated. So if we have an enzyme that will cut only in the absence of methyl groups, or vice versa, we can do an experiment. We get DNA from organisms, we cut with this enzyme. The ones that are uncut, we say are methylated. If we know the enzyme is inhibited by methylation on DNA, if it cuts, we say it's unmethylated. So we can do the exact analogous thing. We can cut to determine whether individuals' chromosomes have different nucleotides. We can cut to understand if different chromosomes have different methylation patterns, and then we can sequence them as well. Does that make sense? So here's the example from your book for the bisulfite sequencing. So some cytosines are methylated, some are not. So that's what it's showing at the very top strand, right? There's also some complementary methylated ones. Guanosine are not methylated, but they are part of that CPG island because they come to those dinucleotides. So if you treat with the chemical sodium bisulfite, you convert the unmethylated cytosine to a uracil. And then in sequencing, right, we're sequencing the complementary strands. We still have to do a reaction where we have primers, and we sequence across. So we have C to U transition here, and then we have U to T. It's not, I don't want to, this is a chemical transition. This is due to complementary DNA synthesis. Right, you saw that in discussion last week, that you're not sequencing the original template, but you can infer something about it by sequencing the complementary template, because they're just complementary anti-parallel strands. Then you compare them. So I'm just going to grab this one because I can see it clearly. So this was the unmethylated cytosine, now it's a tyrosine. So comparing the sequencing result of treated and untreated DNA will tell us which cytosines were methylated. It'll tell us whether the gene is methylated at all, whether the gene is differentially methylated in different individuals with different phenotypes, right? It's exactly parallel to the information we want to gain from these experiments to understand nucleotide changes. We can do everything exactly the same to understand methylation changes. Questions on that? So I mean, there's a ton of people who do this, grad students who just like, oh yeah, we bisulfite sequence stuff. But you should think about how, why does it work? Someone had to understand the chemistry enough of DNA to say, well, I think we can do this, right? That's the hypothesis. It's still science. Research and development is still hypothesis testing. I hypothesize that if I treat DNA with sodium bisulfite, I can convert things, right? Chemistry in your intro chemistry labs might not feel hypothesis testing, but it is at the heart of it, right? It's still science. Okay, that's enough with the DNA side of things. Let's scale up to these histone side of things. We're not going to talk a lot about them, except they're highly variable. So there's more than 100 different kind of post-translational modifications to histone proteins. So that's the key point here. Histones are being modified post-translationally. That sort of makes sense to me, in the sense if we're saying DNA sequences are the same, we're not generating different types of histones, though there are many different types of histones. We're changing histones. If a certain type of histone is going to bind in a certain region of the genome, we don't want to alter that. We want to alter, the cell is altering the modification. So the one we talked mostly about was acetylation. There is methylation though. There's also phosphate groups. What's important maybe about phosphate groups? Any ideas?
That's the backbone of DNA.
Can you say it again? Yeah, right. Acetyl groups, methyl groups are non-polar. They're going to have very different properties on histones than polar modifications, like phosphates. At cellular pH, you often see phosphates with those really strong negative charges like oxygen groups, same as you see in 5' ends of DNA. I forget, ubiquitin is just like a big carbon ring, right? I don't remember. But if it's a big carbon ring, aromatic rings have different chemical properties than kind of methyl groups. So that's not that you have to memorize these groups, but you have to think about it. If you want to change histones and you only have so many ways to do it, how are you going to change them? Methylation, add a big carbon ring, or add something polar. Okay, the maintenance. It's really analogous. Like methylation had to be transmitted through successive rounds of DNA synthesis, through semi-conservative. Histone modifications need to be passed down through daughter cells in cell division. Your book doesn't talk as much about this, and I think it's just because we know less about this. But we know it has to happen. We can detect differences in these histones through subsequent cell division cycles, and we know how cell division works, so there must be proteins that are kind of transmitting this information through successive cycles. The only time where this doesn't really happen, right, daughter cells, that's a specific term we use when cells are identical. So in gametogenesis, where you're getting different haploid products, right, sperm egg, this is a very different process. There still is histone, there still is chromatin structure, it's very different because they're not daughter cells. Right? A sperm cell is not a daughter cell, it's a germline stem cell. It has a different process. So at the cell level, how are epigenetics shaping phenotype? So there's really just two slides on this that I want to think about, and that's really about cell differentiation. You can care about the whole organismal phenotype, right? Queen versus worker, but at some level that has a cellular component. Cells have to differentiate. Workers have atrophied ovaries, queens are egg machines, right? They're undergoing different tissue morphogenesis. So how do they do that, right? So just some terminology that your book was talking about. So first we have stem cells. So these are undifferentiated. Stem cells are often described, even though I don't think this is 100% the case in all stem cells, but oftentimes we describe stem cells as being pluripotent. That's the ability to differentiate into any type of cell. They kind of go hand in hand. I have to take a cell, I have to differentiate it, so I'm just going to have a word to describe it, that capacity, pluripotency. And then we have induced pluripotent stem cells. So we've seen induced before in terms of gene regulation. It's the same thing, right? Inducible, it's off and it gets turned on. In the terms of pluripotency, it's undifferentiated, and we're going to force it to differentiate. So specifically, this is not happening from like naive stem cells. So this is when an adult somatic cell. So right, adult, daughter, the terminology gets a little weird, but it's meant to be a cell that's already differentiated. So it's forced to take on this pluripotent state again. So sorry, that's really messy, because I was not gonna spell de-differentiate correctly. So we can get the sources. This is why methylation in epigenetics is really interesting. It doesn't have to happen once through development, from stem cells to tissue. It can happen in subsequent interactions with the environment, because adult somatic cells, so again, somatic or like non-germline, can de-differentiate. They can go back into that stem cell-like state, be pluripotent, and then differentiate again. So we're not going to talk about it a lot, but it's pretty, in my opinion, I'm not a developmental biologist, but this is the most interesting part of this to me. And it's not like an end, it's not like hard stop, you're a liver stuff. Sometimes cells can do weird stuff and go back to being stem cells. So what does that look like? So this is a figure from your book. You get adult cells. There has to be something converting them. What's going to convert them? Transcription, right? So that's why there's transcription factors. You know, we've seen that the dogma is DNA to protein, but there's always that intermediate step of transcription. Transcription is so important to gene regulation. So we're going to go back to these iPSCSs, induced pluripotent stem cells, and then redifferentiation. So here is de-differentiation. And then you differentiate. Okay, so you don't have to memorize any of this, but these terms you're going to be seeing when you start doing your reading articles for developmental genetics. And so I didn't mention this in the beginning of class, but I think you're done with your reading homework now, right? Yesterday was the last one. So we'll see new material next week, but there won't be an additional reading homework. So you read last week, you should have read genomics, genetic applications, epigenetics. This week you read developmental genetics, and I think it says immunogenetics, which we'll talk about very, very briefly, cancer genetics. So you're done with reading. Okay. So the three things we started with in the beginning of class were we're going to methylate DNA. We talked about that. We talked about how important DNA methyltransferases are for the system. We talked about how we can make a logical connection and think about the connection to phenotype. We talked about that now at kind of the histone level. The main importance of that is that it's happening at a cellular level. So cells can contribute to phenotype through the pluripotency. And now we're just going to do the last example, RNA molecules. RNA molecules are not always templates for transcription. They can have catalytic or different functions in the cell, one of them being epigenetic function in this very specific context of X inactivation. Probably in many other contexts as well. This is just the most well-studied one. So we think if this is true, then other systems could be regulated like this. But to my knowledge, I don't think we have good examples. But this is our really best example of this. So what is the phenomenon of X inactivation? So an XX individual, one of the X chromosomes is silenced. So your book says permanently, nothing in biology is permanent. If it helps you to think about that, that's fine. Permanently silenced. You inherit two X chromosomes, but often in a cell, you're only expressing one of them at a time. How is this occurring? How is something having a specific effect on an X chromosome? It's through the action of a lot of genes and what's known as the X inactivation center. Linked genes are physically located in the same part of the X chromosome. They encode what are called long, non-coding RNA. Why do you need all of those kind of adjectives on the RNA? Because we've seen RNA that gets transcribed. We've seen So non-coding takes care of that. It's not gonna be, or sorry, translated, it's not gonna be translated. So non-coding takes care of that. We've seen a lot of short RNAs that are involved with different proteins to recognize transcripts or DNA and silence transcription. So these are long. They're still gonna have similar effects on transcription, but not through the same mechanism, not through those, what did we see? Was it RISC-2 that we saw in class? So that's why we need those two additives. Non-coding is not gonna make the protein long, it's gonna go through different pathways, not through the RISC-2 dicer, cut them up really small pathways. So this is what it looks like in these books. So it's kind of labeling things backwards. So it's saying, okay, we know that one of these X chromosomes is going to be active, one of these X chromosomes is going to be inactive, so it kind of labels them that way, so you can follow the fate of these X chromosomes. So there's the exist gene. So this locus is X-linked. I don't think it has to be X-linked. I think it's hard to do these experiments because they're only happening in kind of like vertebrates, but I think, I might be wrong, but I think you can change the location of this and still see the phenomenon. So why it's on the X locus, something through evolutionary history selected for it, or maybe it was drift, right? It's important that it's on the X now for how it's inherited, like now. But I think if you had to build a system like this from scratch, I don't think it has to be X. Okay, so RNA is transcribed for only one, right? And it coats the one that's going to be inactivated. So when it means coats, how can RNA interact with DNA? It's not opening up the DNA and making complementary interactions. If that's where your brain goes, I think that's fine. That's how we described RNA for a really long time. But it can interact with other stuff, right? RNA can make loops, they can have polar parts, so it can interact with different proteins on the exome. So we named that RNA, the exist RNA, based on the structure of the gene that's been transcribed. And then these are histone modifications. So this is one type of histone modification. It's important that it's a specific type of histone modification because different other enzymes recognize histone modification. There's like H3K9, there's a lot of different types of histone modifications. So it's important that this is the specific one. So what does recruit mean? Like those are terms that molecular biologists use when we don't really know the mechanism, but we see patterns. So this recruit means they kind of co-localize. So you do an experiment maybe where you delete the exist gene in mouse, and you never see these proteins come to the X chromosome. So that's what recruit means. That long non-coding RNA is required for this protein to localize to the X chromosome. I don't know if you know how or why. Also interestingly, like from an evolutionary standpoint, why would you want to, like this is gonna anthropomorphize an X chromosome, but why would you want to silence yourself, right? That's kind of a weird, strange thing too. I think there's reasons you can come up with. We're not gonna talk about them, but if that's something you're interested in, we can think about it. Well, why would you silence yourself? Maybe you have to because of dosage differences, but still, like why would it not be stochastic? Why is it always the X chromosome that transcribes exists is silenced? And so this figure in your book is just to show that, they're physically on top of each other. So these are the 4 genes that are essential for X inactivation, right? They are all located similarly in the same region of the chromosome. Why is this the case? Can you guys think of any ideas how kind of this system built over evolutionary time might have resulted in these being in the same location?
Polarity.
Or what's a feature now that they're together? What's unlikely to happen? Exactly, right? If all 4 of these are required, if they're all really tightly linked, recombination's not gonna break them up. We've thought about recombination for identifying genes, but recombination's a natural process, right? Sometimes recombination can be beneficial and bring genes that are not on the same chromosome onto the same chromosome. It can increase fitness. But it can also have the opposite effect. If genes are required to be together for transcriptional purposes, whatever, if you're breaking them apart, you're gonna inherit now X chromosomes that don't have all the functional components of X in activation. You transcribe 2 X's, things go wrong. So that's very it's a bad argument, but I think hopefully it illustrates what I'm trying to say. Okay, so X in activation is really similar to the topic I wanted to talk more about. So X in activation is happening specifically on the X chromosome, but we have this similar principle of kind of silencing one allele or the other from a specific parent called genomic imprinting. So this is caused by epigenetic differences inherited from different parents. So we've seen this before. We'll see a slide of it. We saw this when we were talking about X chromosome, or we were talking about sex chromosomes and inheritance of sex chromosomes. We said here are how Y chromosomes are inherited, here are ZW chromosomes are inherited, then we see all these extra things. We saw like sex-limited expression, sex-biased expression, then we saw imprinting. So we saw it in that context, but now we can think about it specifically in this context. How are alleles going to be transmitted through gametogenesis and have different methylation marks? So in your book, they talk a lot about consequence of this, thinking about kind of genetic conflict. We're not going to talk about that. What I want to talk about instead is, given imprinting, how can we predict phenotypes? So what would that look like, right? You'd have a chromosome like this, that's a methyl group, in one parent, and the other parent, no methyl, and then you have the zygote. What's going to happen in this F1? Is it going to inherit the methylation mark? Is the methylation mark going to be taken off? Well, it depends on the parent that it inherited the allele from. So that's what we want to think about for this next slide. So here's the example in your book. So they're showing kind of this A1 protein and Y is like an allele, right? We've seen many different ways to write alleles. The plus is always wild type. The plus does not always mean dominant, so remember to be careful about that. And they're showing this 1. So you have 2 parents that are one parent that's homozygous, one parent that's heterozygous with this mutation, but it has different properties depending on the parent that it's inherited from. So let's just start on the right-hand side. So when the mutant allele is inherited from the XX parent or from the egg parent, it's silenced. So what does that mean? It must mean that this F1 here, inherits the methylation pattern. There can be active silencing. There could be some function of that A+ allele that silences. That kind of takes way too many steps. We know that methylation has to be consistent in a chromosome, in DNA strands, right? So right, these chromosomes are independent. They undergo their own DNA synthesis, right? They're not forming a 2-stranded chromosome. But there's going to be consequences for having different methylation on different chromosomes. So maybe there's just a methylation pattern that's being inherited. And I don't think we know exactly how. We know how it happens in DNA synthesis, but through different chromosomes. I don't think we know how, but we can imagine ways, right? We know DNA, different chromosomes, homologous chromosomes line up at certain parts. We know that they recombine. There's ways that we know chromosomes can interact. So this would be the inheritance of a methylation pattern. I think that's the most easiest way to explain that, because it's going to stop transcription of that gene, right? Or inhibit transcription. I'm not saying that's the only mechanism, I'm just trying to provide one. We've been talking about methylation, so methylation can happen. In the flip case, where the mutant allele is inherited from, so right, the XY parents now, they didn't write them in the same direction, it's expressed. So maybe the methylation pattern is inherited differently. So what do you need to be able to do with something like this? So this is the key point for this kind of section of the topic. You don't need to know the details of axon activation, but given different methylation patterns, if we say that one allele is imprinted through the XY parent, be able to do 2 crosses and predict the phenotype. Does that make sense? We're not going to practice this, I don't think, in discussion. But if you want to practice it, we can. But think about this. It's pretty simple Mendelian genetics, right? What's the frequency of inheriting being a heterozygote? 1/2. Well, what's the phenotype? You can't just look at the genotype. You have to know how this methylation is being transmitted through parents. Right? So you take Mendelian proportions. And then you take into account imprinting. And together, these give you the phenotype. So the last slide about epigenetics, most of what I've talked about, hopefully you can see how it's connected, kind of these changes and modifications to DNA, chromosome, protein. But then this is, I think, where people think of epigenetics as being like these magical things. Because then all of these other processes that are really fascinating get thrown into epigenetics. So here's a study from your book. So young rats, they've done an experiment where they allow, like, why do animals groom each other? Or what is their functional purpose in, like, social grooming? We know that's true for apes. What about in rats and kind of things like that? So they do these experiments, and this is like the take home from this experiment. So young rats exposed to more licking and grooming develop different patterns of methylation. So what's that going to happen? That's going to alter gene expression, right? And they found that it actually alters genes important for stress response. That's so weird, right? Like, but you can make predictions. If there's a phenotype of rats that receive grooming and not, how did that happen? It had to happen in changes in gene expression. How do we get differences in gene expression? Differences in methylation. I don't think we know yet. The book's only a couple of years old, 2017. So maybe in the past 5 years, someone has figured out why long rat licking changes methylation. But either way, right, we don't need to know all the pieces to make predictions. Gene expression, there must be a difference in methylation. We do an experiment where we allow a mother rat to groom or not groom, and we see the change through bisulfite sequencing, right? So we can start building this model. So I'm just going to try to write this model explicitly. So you get this pain phenotype, right? That must have to do with different expression. That's an easy way. We know how expression leads to phenotype. Now, how do you get differential expression, right? Methylation. The thing we don't know, but we can experimentally show, is grooming. So somehow grooming leads to methylation changes that lead to gene expression changes that lead to this pain behavior. Okay. Any questions on that? This is just one example, right? One example of how you can build a logical model based on all the information that you know about epigenetics. Okay, the rest of the class we're going to do some developmental genetics. We'll revisit developmental genetics on Thursday. So this is just trying to introduce developmental genetics, provide a couple examples to motivate what you're going to do in discussion tomorrow. So this was supposed to have a picture and I just forgot one. So we're transitioning. We're done with epigenetics, we're going to developmental genetics, but really they're kind of gonna be interrelated, right? If developmental genetics is about transitioning or differences in morphology, we know epigenetics can play a really important part. So what kind of phenotypes do people care about for developmental genetics? Often, it's not the phenotypes we've seen previously, like black coat, white coat, not these binary things, but more kind of quantitative traits. And the reason that developmental geneticists care about those more is because it's not about kind of laying the foundation of a body plan, but it's changing it. So that's what's happening here. So these are two fish called sticklebacks. So this is looking at the fish, I think, from below. Looks like if the fish was on the back. And they have these spines. So we're not looking about the presence and absence of spines. If we found mutants with no spines, that wouldn't tell us about development. That would tell us about something way kind of farther up in embryogenesis. We want to think about development. So spines are there, they're differential length. So something must be happening. So one of the models for developmental biology is fruit flies. You don't have to memorize any of this, but we want to think about like, what is this telling us? It's telling us that we can look at something like development in this fly. We can think about genetic changes that control developmental changes. That's the main take-home point for this part. So all of these slides are just to build up like why use fruit flies in the first place? They're kind of weird little things. Why use them? One is because they go through really rapid cellularization. So this is embryogenesis. You don't have to read all of it. The main take home is rapid cellularization. Humans take a really long time to develop. Fruit flies, all of this is like a couple days. So they're gonna set kind of their body plan up based on how nuclei are migrating in the cell. This is pretty similar for lots of organisms, not just insects. Most organisms, you have to have a polarity, right? If you think about humans, we have a head and a feet. Sometime during embryogenesis, that polarity, that gradient, had to be developed, right? You have bilateral symmetry. That line of symmetry had to be developed. So even though the exact genes are not going to be the same in fruit fly and invertebrates, the principles are the same. You have to, through development, set up boundaries of expression, gradients of expression, and domains of gene expression to give features that are going to come out in the adult animal. So this is what this is trying to describe. Really early on in development, right, that embryo does not have a head. It doesn't have an abdominal segment, but the genes that are there, localized through differential gene expression, are going to stay there, basically, throughout development. So when you look at a figure like this in the book, like, who cares? What does this mean? That's a take-home point. A major body axis has to be defined. How are these genes going to differentiate? Through different regulation of gene expression. Right? All these things we've already seen. So, kind of segue before we get into our last clicker question for the day. Well, how is that happening? Right? How is all of these, like, this has to happen really, really quickly. How? It's not all from the embryo itself, interestingly. Lots of these age polarity genes. So polarity refers to not like chemical charge, but like north-south pole, like orientation. Egg polarity genes, a lot of them actually have maternal origin. Eggs, whether they're Drosophila eggs, plant eggs, right, seeds. Human embryos are pumped full of RNA and protein. So that would make sense that some of them have a function. So the maternal origins are really important for determining anterior posterior axes. Those like front and back, and dorsal ventral. So dorsal ventral. So anterior, posterior, dorsal, ventral. Those are like four axes, right? You give now this embryo kind of has like north, south, east, west. It has a polar coordinate system that can be used to build everything else. Since this is the very first kind of axes that have to be laid out in this embryo, it makes sense that they might have maternal origin. The mom kind of gives the head start to these embryos through development. Here's your first genes that are going to establish these axes. And then the rest of them are what we call morphogens. These are mostly transcribed by the zygote itself, not always, and they're concentration gradients. And that gives kind of developmental phases. What does that look like? I'm going to skip this one to this one. So here's an example. So this is a gene called licoid. You can label it. We talked about how can you localize RNA in a cell. We talked about that. You do an experiment called an in situ experiment, right? You create an RNA molecule that can hybridize with other RNA or DNA that hybridizes, right? It physically interacts. You take these embryos, you fix them. Fixing means you make them chemically, you change them chemically so that you can insert RNA in through the cellular membrane. You have an RNA that's labeled with fluorescent dye, it interacts through complementary interactions. You look under a microscope and you can see it. So this is one example. So bicoid is important, it has a gradient. So high expression in the anterior, and then it tails off. But the absence of bicoid doesn't give you posterior. So I'm just going to try to write that. This is really important. It's in many systems in developmental genetics, it's not presence or absence that matters, it's these gradients. So to get a posterior, if you had a bicoid mutant, you wouldn't like now have posterior everywhere. You would have failure to develop an anterior segment. So these would actually be embryo inviable. Instead, there's another gene, nanos, which is in the posterior. So you have kind of like complementary gradients in gene expression. So the nanos is a little harder to see right there. It's a much more limited expression. So what I want you to be able to do with this type of information is read these types of graphs and think about if there were changes in these types of graphs, what would that tell you about the DNA genetic changes? So this is our last question, but then we want to kind of take this one step further. So read this type of graph. What does it tell you? So there's a high concentration of which protein to stimulate anterior structures. So I just told you, but hopefully you can read this and figure it out. Okay, so as you're answering this, I'm going to draw something. So let's say now we have a mutant. We found this bicoid mutant that now, instead of being expressed in this kind of localized region in the anterior, it's expressed in the whole embryo. So the best answer was bicoid. So now we have a mutant with increased expression. What's the most likely DNA change that happened here? Where in this bicoid gene or next to the bicoid gene did this mutation occur? I'll tell you it's a cis mutation, because I know some of you are like, well, I don't know if it's cis or trans, right? So eukaryotic gene expression, what do you need for gene expression? So, I don't think we're practicing this in discussion. This verbatim is on the exam. So what kind of mutation? So I'm not gonna answer it. You guys can figure it out. You can get back to me about that. What type of mutation probably led to the increase in expression? Think about what is required in eukaryotic genome expression, what's in cis with the protein coding genes. I didn't tell you the protein coding genes there. So what's most likely to have changed to increase expression? Okay, so that's all we have for today. It's like dead silence. I'm not answering this. You guys figure it out. Talk to me in discussion or Dr. Drew in discussion tomorrow.